Okay, so good day. My name is Kenneth Maynard. I work at Sanofi Aventis in the US. It's one of the top five big pharmaceutical companies. And my responsibility is to look for scientific external innovation and to partner with institutions, with biotechs, with other big pharma to bring in some of that scientific innovation into a new therapeutic strategic unit focused on aging. The presentation that I gave today focused on the impact of the concept of hormesis, U-shaped or bell-shaped dose-response curves in pharmaceutical R&D. Over the last 15 years, we've seen an explosion in the level of information around hormesis across a wide variety of medical science as well as clinical development. And the impact on this is that it really focuses our attention on how well we do at deriving dose-response information for our clinical studies, as well as how well we obtain concentration response data at the level of the bench and in research. The presentation focused on trying to determine what we need to do in the pharmaceutical industry to incorporate our understanding of hormesis so that we can have a more positive impact on more positive clinical trials and to gain a shorter time frame for R&D and ultimately reduce the cost of R&D for drugs and biologics. So it is clear that in many circumstances, trials are stopped because there is adverse events, severe adverse events, or trials are completed and they are found to be not efficacious in a fairly late stage of development, in phase 3, for example, when you have already spent about 10 to 12 years developing this drug and round about $800 million to $1.2 billion, depending on who you read. Part of this may be simply because we've chosen the wrong dose to test in critical pivotal phase 3 studies. By using the concept of hormesis and understanding that going into our R&D studies, that many of these dose responses or concentration responses are not simply linear or threshold, but they can be U-shaped or bell-shaped. It forces us to look at a more thorough investigation of the dose response and ultimately better designed clinical trials. Today, I focused on 2 or 3 different solutions at different stages of development that could allow us to gain better insight into concentration response and dose response information, since clearly what is required is for us to sample often, starting low, which requires us to do more doses and longer studies. But there are technologies and there are techniques available today that will allow us to do this in a cost-effective manner. At the level of phase 0 or microdosing, for example, we can use technologies such as mass spectrometry and PET imaging to look at microdoses. And using this microdosing technology, we will be able to find out whether or not the dose response curve using very small amounts in humans actually work at the level of the cell. And we've seen now over the last 5 years that these data correlate very well to the ramped up human response data, so the impact of the dose at the whole body level. So there's microdosing. And in the phase 1 level, there's this concept called pharmacometrics, which is a combination of looking at the drug, looking at the disease, and looking at development, looking at clinical trial data, and modeling it so that you really optimize your understanding of pharmacokinetics, what the body does to the drug, as well as pharmacodynamics, what the drug does to the body, in order to thoroughly understand dose response information and get a better idea of the doses that we should be using in later phases of the clinical trial. In later phases of the clinical trial, phase 2 of the clinical development, phase 2, phase 3, there's the concept of using more powerful and more flexible statistical design, such as adaptive trial design. Adaptive trial design allows us to interrupt the study and have an analysis of the data and change the parameters of the trial, whether it be using originally a biomarker to try and define a responder group, which could then be used in your phase 3 study. Adaptive trial design also allows us to look at phase 3 studies in such a way that you look at drop doses that are not effective and hone in on doses that could lead to positive outcomes in phase 3 studies. The final aspect of the presentation that I gave today simply looked at the International Conference on Harmonization, which happened in the mid-1990s, and it's a way for different regulatory bodies in the United States, in Europe, and in Japan to get access to data that would allow the marketing authorization across different countries using essentially an agreed standard to obtain clinical trial data. And the recommendations that they make in their 1994 document for dose response studies very clearly indicate that studies should try to use the entire database of information that you obtain during clinical development to obtain a clear view and understanding not only of the shape of your dose response, but also the timing of the response. There is a concept called temporal hormesis, which actually was shown very nicely in studies looking at beta blockers in chronic heart failure. And in that case, it was not necessarily just the shape of the curve, but also the timing of the curve, so that when you take beta inverse agonists, the benefit of that for chronic heart failure patients is not seen the day after, but it's actually seen the month after or 3 months later compared to ACE inhibitors. So there are ways now that are very clearly defined, partly by the International Conference on Harmonization. Sponsors of clinical trials should now be using very carefully these International Conference on Harmonization recommendations and guidelines to obtain clear, well-defined clinical trial dose response data, and can use techniques and technologies such as microdosing, such as pharmacometrics, and such as adaptive trial design to help us get there. And ultimately, I believe that this will result in better studied drugs and hopefully more effective clinical trials, resulting in more drugs on the market for our patients.